Hello, my name is Dr. Newmaster. I'm a professor of genomics at the University of Guelph for 20 years and actively making molecular diagnostic tools. And today I want to talk to you about some of these genetic molecular diagnostic tests that we use for detecting COVID. And there are several ones. There's the PCR-based RNA test that's based on genetics. There's the antigen test and the antibody test. And today I'm going to talk about the antibody test and why it is perhaps at this point in time for us quite fit for purpose. So, what's the question at hand? Do I have some protection, some resistance to COVID-19? And we get that several different ways. A natural infection where your body then produces antibodies, or you get vaccinated and the body produces antibodies. The antibody test is based on several different designs, so not all antibody tests are the same. Some antibody tests are based on the structure of the virus, which you have probably seen before, which is made up of spike proteins, nucleocapsids, and a couple of other structures on it. For vaccination, when they produce a vaccine, they base it on the spike protein. And that spike protein then induces antibodies, IgGs long-term, IgM short-term, for the spike protein. So if you had only a test for the spike protein, and you had an antibody test that was for the spike protein, it would show you have antibodies after vaccination. If you had a natural infection, the natural infection would produce antibodies based on the nucleocapsid. The nucleocapsid would be very important in designing an antibody test so that we would know that you produced antibodies, IgGs and IgMs, to natural infection. And the problem is if you only have a test with either an antibody test that is designed based on spike or nucleocapsid, then you wouldn't really know if you had antibodies produced from either one of those events. So the best thing to do is get a test that has both. That way, if you had a natural infection or you had vaccination, you would be able to know that you have antibodies. You would know when your antibodies are gone because you would have a negative test. And of course, we're all going to have the question, when are my antibodies going to be gone in case I'm exposed to another virus, another strain of COVID? And will I actually produce antibodies to it? And this test will be able to do that.